Okay, good afternoon, everyone, and welcome to the second session of the Expert Reviews in Molecular Medicine author webinar series. Expert Reviews in Molecular Medicine is an online journal featuring authoritative and timely reviews on all aspects of molecular medicine. Review articles cover current and emerging understanding of the molecular mechanisms underpinning human health and disease, and molecular aspects of the approaches used to diagnose and treat them. Nicola Curtin is the new editor-in-chief of the journal and has been working to refresh the scope and appoint new editorial team members. In a moment, I'll hand over to Nicola, who will be introducing today's session with Hannah Smith and Harriet Southgate, our guest speakers. So to kick off, I'd like to invite Nicola Curtin, editor-in-chief of Expert Reviews in Molecular Medicine.
Thank you, Ali. It's my great pleasure actually to introduce Hannah and Harriet. I'm really proud because they were both my PhD students, and the reason they're giving this seminar today is not because they were my PhD students, but because they co-wrote an article for Expert Reviews in Molecular Medicine that was published in 2020 and has been very highly cited since then. I think it's an interesting topic that they are talking about, and they're writing a publication during their PhD was not only an added bonus to their CVs, but also helped them with the writing of the introductions to their theses. Harriet is now working at AstraZeneca, but what she's going to be talking about today is purely studies that were undertaken while she was in Newcastle. And Hannah Smith is still in my lab in Newcastle. So I'll proudly hand over to them, and I will handle any questions that arise from this talk.
Great, thanks, Nicola. Let's get going then, I guess. Sorry, give me 1 minute. Start again. There we go. Right, yeah, so thank you very much for that lovely introduction, Nicola. I'd like to thank also the organisers for inviting us to give this webinar today. Like Nicola said, our review on the DNA damage checkpoint kinases in cancer was published last year, and we'll just give a quick overview of the review paper for you today. These are my disclosures, like Nicola mentioned. So I'll start by giving a quick introduction into the role of the DNA damage checkpoint pathways, and followed by discussing the role of the ATM CHEK2P53 pathway in cancer and the development of inhibitors targeting it. I'll then pass over to Hannah, and she will discuss the role of the ATR CHEK1V1 signaling pathway and the development of inhibitors targeting this pathway, and give our perspectives on targeting the DNA damage checkpoints for the treatment of cancer. So the DNA is constantly under attack from endogenous and environmental damaging agents. So a signaling network to identify and repair the damage and arrest the cell cycle is required to prevent the damage being passed on. Therefore, the DNA damage response is essential to maintain genome integrity and for cell survival. ATM and ATR are DNA damage sensor kinases at the apex of cascade signaling to both cell cycle arrest via the inactivation of cyclin-dependent kinases and to DNA repair. ATM and ATR have distinct and overlapping activities with substantial crosstalk between the two pathways. And our review focused on the role of these pathways in the DNA damage response and the development of drugs targeting them for anti-cancer therapy. So the first part is discussing the role of the ATM CHEK2 p53 signaling pathway and how it could be targeted to enhance cancer therapy. So ATM is activated in response to DNA double-strand breaks, and recruitment to the breaks by the MRN complex leads to ATM activation. Active ATM then activates CHEK2, which together with ATM signal to promote DNA repair. CHEK2 inactivates CDC25A, preventing cyclin-dependent kinase 2, cyclin E activation, and the G1S transition. ATM and CHEK2 also activate the tumor suppressor protein p53, which upregulates the transcription of the cyclin-dependent kinase inhibitor p21, preventing CDK2 cyclin E activity and reinforcing the G1 cell cycle arrest. So aberrations in this pathway are commonly observed in cancer. Homozygous germline mutations in ATM results in ataxia telangiectasia, which is a well-characterized recessive genetic disease, which predisposes to cancer. Somatic mutations and loss of expression in ATM is also observed in many cancer types, including colorectal cancer, breast cancer, non-small cell lung cancer, lung adenocarcinoma, and pancreatic cancer. ATM loss of heterozygosity also occurs frequently through deletion of the long arm of chromosome 11. 11q deletion is commonly observed in breast cancer, chronic lymphocytic leukemia, and other lymphoid malignancies, and in childhood neuroblastoma, where it predicts poor survival. Homozygous germline mutations of p53 or CHEK2 manifests in the cancer predisposition Li-Fraumeni syndrome, and somatic CHEK2 mutations are also observed across cancer types, although these are much less common than mutations in ATM. And p53 is the most commonly mutated gene across all human cancer types. Somatic p53 mutations are observed in around 40% of all tumors, with the mutation rate varying between cancer types. As with ATM, p53 loss of heterozygosity through deletion of chromosome 17p is frequently observed in cancer, and loss of 17p is often accompanied by mutation of the other p53 allele. So the rationale for the development of inhibitors of this pathway is that germline ATM mutations in humans results in ataxia telangiectasia, and these patients are very radiosensitive. ATM null mice are also radiosensitive, so targeting ATM with small molecule inhibitors should sensitize cells to radio and chemotherapy. In contrast to ATM knockout mice, CHEK2 knockout mice are radioresistant, and the context in which CHEK2 inhibitors will have therapeutic benefit remains unclear. So the first described potent and specific ATM inhibitor, Q55933, was developed by Kudos Pharmaceuticals in collaboration with Newcastle University, and was shown to enhance the cytotoxicity of ionizing radiation and topoisomerase 1 and 2 poisons in vitro. But its poor aqueous solubility and in vivo bioavailability precluded any further preclinical testing. Another ATM inhibitor was identified by Pfizer and showed similar radiosensitizing properties to Q55933. However, the short half-life of this compound in mice prevented any in vivo studies. And in 2013, 2 structural derivatives of Q55933 with improved potency and aqueous solubility were reported. And although the pharmacodynamic properties of these compounds were still not suitable for oral administration, Q600919 effectively radiosensitized glioblastoma in vivo when directly injected into the tumor, and Q59403 sensitized tumors to ionizing radiation and topoisomerase 1 and 2 inhibitors in vivo by intraperitoneal injection. And in 2017, GlaxoSmithKline identified a novel radiosensitizer from a screen of published protein kinase inhibitors, which was shown to act through inhibition of ATM. And importantly, this compound showed an additive radiosensitizing effect when combined with PARP inhibitors. In 2018, AZ32 was reported by AstraZeneca to be an orally bioavailable inhibitor of ATM, which is capable of crossing the blood-brain barrier in mice. Optimization of this compound led to the development of AZD1390, which sensitized brain tumors to radiotherapy in preclinical models, justifying translation into clinical trials. And AZD0156 was also developed by AstraZeneca following the optimization of a different lead scaffold. And this compound showed good pharmacodynamic properties and synergism with topoisomerase 1 poisons and PARP inhibitors in tumor xenograft models. AZD0156 has also entered a phase 1 clinical trial. And finally, N3541 was reported by Merck and also sensitized multiple tumor cell lines to ionizing radiation, topoisomerase poisons, and radiosensitized tumor xenograft models. This compound has also entered a phase 1 clinical trial. And many CHEK2 inhibitors have also been developed. And in general, CHEK2 inhibitors show modest anti-proliferative effects when compared with ATM, ATR, or CHEK1 kinase inhibitors. And they have shown to be radioprotective, but have also been reported to potentiate the cytotoxic effects of gemcitabine, cisplatin, topoisomerase poisons, and PARP inhibitors. So the ATM inhibitors M3541, AZD0156, and AZD1390 are currently being tested in clinical trial in combination with radiotherapy or chemotherapy agents, but no specific CHEK2 inhibitor has progressed into clinical trials. So to summarize this first section, the ATM CHEK2 P53 pathway is triggered by double-strand breaks and signals primarily to G1 cell cycle arrest and DNA repair. Mutations in ATM, CHEK2, and P53 are associated with cancer development. And targeting ATM with inhibitors sensitizes cells to radiotherapy, topoisomerase 1 and 2 poisons, and PARP inhibition. ATM inhibitors are currently being evaluated in clinical trials. CHEK2 inhibition is associated with radioprotection, but may sensitize to topoisomerase 1 poisons and PARP inhibition. But no CHEK2 inhibitors are currently being evaluated in the clinic. So I'll now pass over to Hannah. She will continue with the second section of this talk.
Thanks, Harriet. So I will start by giving an overview of the ATR CHEK1 and WEE1 pathway, followed by dysfunction of the pathway and how we can exploit this in cancer cells. So the ATR CHEK1 WEE1 pathway is activated in response to replication stress and DNA damage. Single-stranded DNA allows for the recruitment of ATR activating proteins. Although this single-stranded DNA primarily arises from stalled replication forks, it is also the result of nucleotide excision repair intermediates and resected double-strand breaks. ATR is recruited by its partner ATRP, which begins the signaling cascade. ATR activates CHEK1, which in turn causes the inactivating phosphorylation of CDC25A and C. This inactivating phosphorylation prevents CDC25A and C from removing the inhibitory phosphorylation on cyclin-dependent kinase 2 and cyclin-dependent kinase 1, respectively. CHEK1 has also been shown to activate WEE1, although this has only been shown in xenopus studies so far. WEE1 activation results in the inactivation of CDK1 and 2. So this signaling results in preventing both S-phase progression and entry into mitosis, allowing time for repair and the restart of stalled replication forks. ATR, CHEK1 and WEE1 have also each shown that they signal to homologous recombination repair. The importance of ATR, CHEK1 and WEE1 is highlighted by the embryonic lethality of each component. Seckel syndrome is a rare autosomal recessive disorder caused by a hypomorphic mutation in ATR. This results in delayed development, but not cancer predisposition. Similarly, mouse studies have shown that even when crossing P53 defective mice with Seckel syndrome mice, this resulted in no cancer predisposition. Upregulation of the pathway is common in tumours and is an indicator of poor prognosis. And ATM defective tumours have shown to be particularly sensitive to ATR inhibitors, and this is now being used as a biomarker in clinic. The ATR CHEK1/WEE1 pathway is a good target in cancer cells for two reasons. The first being the involvement the kinases have with cell cycle control. Normal non-tumour cells have G1 and S/G2 checkpoints intact, so when damage occurs, cell cycle progression will be delayed, as described earlier, and the damage can be repaired, resulting in cell survival. When these normal cells have S/G2 arrest inhibited by ATR CHEK1 or WEE1 inhibitors, the cell can still survive by having G1 checkpoint control. P53 mutations are a common cause of loss of G1 checkpoint control in cancer cells. When damage occurs, these cells are only able to survive by relying heavily on S and G2 checkpoint control. When this S and G2 checkpoint control is inhibited by ATR CHEK1 or WEE1 inhibitors, cell death occurs as cells lose all cell cycle control. Targeting these cancer cells that already lack G1 checkpoint control with ATR CHEK1 or WEE1 inhibitors will selectively kill tumour cells, whilst normal cells with intact G1 checkpoint control will survive. As well as their key function in cell cycle checkpoint control, ATR CHEK1 and WEE1 also have key roles in DNA repair. As replication lesions occur and replication stress signals to ATR CHEK1 and WEE1. Each of these signal to key components in the homologous recombination repair pathway, as shown here. With this signaling to RAD51, BRCA2, and cyclin-dependent kinases, homologous recombination repair can occur and the cell survives. However, when these kinases are inhibited, signaling to key HRR components cannot happen and HR is compromised, rendering cancer cells sensitive to DNA damaging agents such as PARP inhibitors. An early study in 1998 found that overexpression of kinase inactive ATR caused sensitivity to DNA damaging agents such as irradiation and cisplatin. This was followed by caffeine being identified as an ATR inhibitor in 1999, and although it lacked specificity, it was shown to inhibit ATR activity at a radiosensitizing concentration. Similarly, a study in 2002 showed that expression of kinase-dead ATR conferred sensitivity to multiple DNA damaging agents, including irradiation and cisplatin. In 2011, NU6027 was the first study of its kind to show that ATR inhibition inhibited homologous recombination repair and increased PARP inhibitor cytotoxicity. Also in 2011, it was shown that the ATR inhibitor ETP46464 had increased cytotoxicity in cells overexpressing cyclin E. This inhibitor sensitized to irradiation and cisplatin, however, it lacked specificity and had poor pharmacological properties. Berzosertib was shown to potentiate platinum therapies, gemcitabine, and irradiation in vitro before becoming the first highly selective ATR inhibitor to go into clinical trials. AZ20 was developed and showed good potency and selectivity in 2013, as well as synergy with cytarabine and gemcitabine. In 2015, a study showed that increased sensitivity of ATR inhibitor VE821 in cells with defective homologous recombination repair and basic excision repair. Elimusertib showed strong anti-tumor effect as a monotherapy in cell lines and in xenograft models before entering clinical trials. And in 2018, ceralasertib was developed from AZ20 and showed excellent pharmacokinetic qualities and sensitized cells to cisplatin, gemcitabine, and irradiation in vitro. Clinical progression with CHEK1 inhibitors has often been halted due to off-target effects. In 1999, UCN01 was identified as a first-generation potent CHEK1 inhibitor. It abrogated G2 checkpoint control and sensitized P53 defective cancer cells to cisplatin, camptothecin, and irradiation. However, it had poor potency and specificity and struggled to bypass radiation-induced G2M checkpoint. In 2008, PF77736 showed potent single-agent activity and sensitized cells to gemcitabine and other DNA-damaging agents. It was found to be more cytotoxic than P53 mutant MYC-driven cancers. Also in 2008, AZD7762, a dual CHEK1 and 2 inhibitor, showed potentiation of cytotoxicity of gemcitabine topoisomerase inhibitors and cisplatin. In 2012, SRA737 showed good selectivity and demonstrated synergy with PARP inhibitors niraparib and olaparib. This compound was taken into clinic both as a monotherapy and in combination with gemcitabine and cisplatin. In 2015, prexasertib was shown to be a specific CHEK1 inhibitor with strong single-agent activity. It has shown synergy in vitro with PARP inhibitors, and this combination has been taken forward into clinical trials. And in 2017, MK8776 was shown to be a potent radiosensitizer, as well as sensitizing cells to other DNA-damaging agents, such as gemcitabine and cisplatin, and this has also been carried forward into clinical trials. Despite the key role of WEE1 in S and G2 arrest, very few small molecule inhibitors have been developed. In 2001, PDO166285 was the first potent WEE1 inhibitor. It radiosensitized cells in a p53-dependent manner. However, limitations included its inability to pass the blood-brain barrier in glioblastoma cells and a lack of selectivity. In 2009, adavosertib was shown to be a specific potent WEE1 inhibitor with excellent selectivity. It was effective as a single agent and acted synergistically, increasing the cytotoxicity of many DNA-damaging agents. Combining inhibitors of the pathway with one another has shown preclinical potential. A study with ATR inhibitor VE821 and CHEK1 inhibitor AZD7762 showed synergistic cell death in vitro and in vivo. Similarly, berzosertib combined with AZD7762 was well tolerated in mouse models and increased survival. One study combined ATR inhibitor ceralasertib and WEE1 inhibitor adavosertib and found this combination inactivated homologous recombination repair and rendered cells sensitive to both cisplatin and PARP inhibitor. CHEK1 inhibitor LY260361 with adavosertib resulted in observed synergistic anti-tumor effects, and similarly, PF77736 with adavosertib showed synergy when used in combination with one another. Preclinical synergy with PARP inhibitors has been observed with ATR CHEK1 and WEE1 inhibitors. ATR inhibitors NU6027, VE821, and AZD6738 have each shown synergy with PARP inhibitors. Similarly, a series of PARP inhibitors synergized with various CHEK1 inhibitors, including UCN01, AZD7762, and LY260361. And fewer studies have looked at the combination of PARP and WEE1 inhibitors. However, adavosertib has been tested in combination with olaparib, which has shown promising synergistic radiosensitization. The mechanism behind this synergy in part relies on the kinases' involvement with homologous recombination repair, as described earlier. When DNA single-strand breaks occur and PARP is present, base excision repair can happen and the cell survives. However, when PARP is inhibited, replication lesions form. Usually these can repair via homologous recombination repair and leading to cell survival. However, when ATR CHEK1 or WEE1 are inhibited, homologous recombination repair cannot occur, and this results in cell death. As you can see from the table above, there are many active clinical trials with ATR inhibitors, particularly berzosertib and ceralasertib in both phase 1 and 2 clinical trials. Noticeably, 9 of these trials are with AZD6738 and PARP inhibitor olaparib, which has strong preclinical support. Many of these trials are now completed, and publication of results is being awaited. Clinical progression with CHEK1 inhibitors has often been halted due to lack of bioavailability and off-target effects. However, CHEK1 inhibitors with greater specificity have now entered clinical evaluation. PF77736 was used in a phase 1 clinical trial in combination with gemcitabine. However, this was discontinued, but this was due to business reasons as opposed to safety concerns. Prexasertib entered clinical trials in combination with DNA-damaging agents. However, there is only one trial with a PARP inhibitor, despite promising preclinical data. Results published from the study highlighted in red have shown that prexasertib monotherapy had modest clinical efficacy. Adavosertib is the only WEE1 inhibitor to reach the point of clinical development. It has been used in a number of trials in combination with carboplatin, gemcitabine, cisplatin, cytarabine, and olaparib. The majority of these clinical trials have been completed and publication of results is being awaited. So to summarise this section of the talk, pathway signalling is triggered by single-stranded DNA, primarily caused by stalled replication forks, and in turn signals to intra-S and G2M checkpoints, as well as DNA repair. Upregulation of ATR CHEK1 and WEE1 in cancer cells is associated with malignancies. ATR CHEK1 and WEE1 is a promising target, as cancer cells defective in G1 control will also then lose intra-S and G2M checkpoint control, whilst normal cells with G1 control remain uncompromised. The preclinical development of ATR and CHEK1 inhibitors has shown good single-agent activity and synergy with DNA-damaging agents, and even though WEE1 inhibitors initially lagged behind, they have now entered clinic. Many in vitro studies have investigated various PARP inhibitors in combination with ATR CHEK1 and WEE1 inhibitors, which have now moved into an array of clinical trials. And to summarise what we've spoken about today, targeting the DNA damage response checkpoint signalling has evolved to the clinic based on sound scientific hypotheses and preclinical data. Targeting the ATR CHEK1 and WEE1 pathway is most promising, as mutations in ATM and CHEK2 are associated with cancer development. ATM defective tumours have been shown to be particularly sensitive to ATR inhibitors, and this is now being used as a biomarker. The most active combinations for each class of inhibitor are ATR inhibitor with platinum therapies and CHEK1 inhibitors with gemcitabine, and clinical data to be published soon can direct future work. Thank you for your attention, and we'd be happy to answer any questions.
Thank you both very much indeed. And while people are thinking of questions, maybe I can start the ball rolling. Harriet, in terms of CHEK2, the protective effects against ionizing radiation and possibly other drugs, is this going to be explored clinically at all?
As far as I'm aware, it has not on the track for clinical exploration yet, but there have been a couple of papers that have described protective effects against the toxicity of topoisomerase, I think just topoisomerase inhibitors and PARP inhibitors in p53 defective models. The sort of the reasoning behind this would be that the p53 deficient tumors would die, but in the normal sort of hematological cells, they inhibiting CHEK1 would rescue those, as they still have functional p53. So it wouldn't trigger that sort of apoptotic pathway.
Okay, and Hannah, in terms of the off-target effects of CHEK1, how do they manifest? Are they like the normal hematological toxicities, or are they other sorts of toxicities?
Yeah, so it pretty much is hematological toxicities are the main off-target effects we see.
And is this, when you say this is off-target, does this mean it's not through targeting CHEK1?
It was found that with MK8776 and the SRA compound, they found that this was mainly due to inhibition of CDK1 and CDK2, as well as
okay. Right, yeah. And am I right in thinking that some cardiotox has been observed with this, or is this other kinase inhibitors I'm thinking of?
I'm not too sure. I'd have to.
Yeah, there is cardiotoxicity associated with CHEK1 inhibitors, which is sort of hindering. Sorry, excuse me. It's hindering their development at the moment, but I think the most leading one is prexasertib, isn't it? That's in the most trials.
Yeah. I had a feeling that there was some evidence there, and obviously people are looking now at immunotherapy treatments, and one of the main reasons that tumours are hot in response and stimulate the immune response is through through DNA damage, classically through mismatch repair defects. What's the role of the DDR checkpoint inhibitors in immuno-oncology?
So I think there's been a lot of talk really targeting the ATR pathway in combination with immunotherapy, but also parts which we haven't talked about here. But with the ATR pathway, there is two schools of thought really. So that the first one being that you inhibited DNA repair, leading to more sort of neoantigen, higher tumor mutational burden, that type of neoantigen expression, which would trigger the immune response. But also there's some evidence coming out about ATR inhibitors downregulating PD-L1 expression, which would also stimulate the immune response against the tumors, sort of enhancing that immune checkpoint blockade with the immunotherapy. The other thing is also with the tumor microenvironment. So sort of there's something to do with T cells in the tumor microenvironment that after when the people or the mice or whatever come off the drug, that leads to greater expression of these immune sort of cells in the tumor microenvironment. It's sort of there's a lot of information coming out about combinations with the ATR pathway and immune checkpoint blockade especially.
And I seem to recall that there was also quite a lot of data about PARP inhibitors. I know, but that was through CGAS sting activation. What's the role of ATR in that as well? Do you know?
Not off the top of my head.
Okay, yeah, it's fine. I just wondered what the evidence was there. And so what do you think are the key differences in terms of the differences between ATR CHEK1 and WEE1 inhibitors?
I think probably the most clinically progressed ones are the ATR inhibitors at the minute. And there's obviously a lot of trials that have now been completed looking at PARP inhibitors with ATR inhibitors. Obviously with WEE1 there was a bit of a lag for them getting to that point of clinical development, but I think adavosertib is a really good compound going forward, and that seems to be in a lot of trials with different
and mechanistically, do you think they're very similar, or?
So they obviously, I guess, like CHEK1 and WEE1 are downstream from ATR, so you might expect that ATR would have more of an impact, but that would depend whether the mechanism of action is mainly through their cell cycle effects or their effects with homologous recombination repair. Because with homologous recombination repair, they each kind of have a key role. I wouldn't say that one has more of a role than the other.
Okay, so it might depend on whatever drug that you were using in combination with them, whether it was something that needed the cell to arrest rather than go down the homologous recombination route.
Yeah, definitely.
I'm trying to see if there's any questions in either the chat or the Q&A thing, and I can't see anything from anybody. Does anybody have any questions from the audience? I don't know whether this is a technical issue or whether it was so clear that nobody has got anything to ask you. I don't know if people can raise their hand.
Yeah, there aren't any questions in the Q&A or chat. So if anyone does have one before we finish, now's your time. And if not, you can always ask them afterwards as well.
Is there an email address that they can send their questions?
Yeah, I'll put my email address, and then I think there should also be a survey going out afterwards to everyone that's attended. But yeah, I'll put my email address here. So yeah, please do email me, and I can forward any questions on to Hannah and Harriet. But if not, I think we could probably end there. And I mean, I hope you've all really enjoyed the talk, and found it really useful to hear from Hannah and Harriet and learn more about the journal. We'd really like to thank Hannah and Harriet for their time and taking part. There are more webinars in this series to register your interest for, so please do check out the information on the Expert Reviews in Molecular Medicine core page. And thank you very much for attending.
Thank you, everybody.